Welcome children. In this video I will be dealing with the topic electron transport chain. We have learnt electron transport chain in two chapters, one in photosynthesis as well as in respiration. So this video is applicable to both the chapters. Electron transport chain, or it is also called chemiosmosis. So I would like to connect it with real life situation, that is osmosis. We know the application of osmosis, and what comes to your mind is this experiment where raisins, dried grapes, are placed in water. After some time, or after 24 hours, what do you see? These raisins swells. They bulge. Okay, so the two raisins have bulged. What has happened? Water has moved from outside the beaker into the raisins. So we saw there water has moved from a region of high water concentration to a region of low water concentration. The concentration of water was very low or negligible, no water inside the raisins, it is dry, but water outside is much. So water entered from region of high concentration to a region of low concentration through a membrane, and this process is called osmosis. So what is required for osmosis? Definitely when we analyze this experiment, a membrane is needed. Second important requirement is a gradient. Water was high outside the raisins. Water was high compared to the water inside the raisins. So a gradient was required. And here what was the gradient? A water gradient. So I am going to incorporate this into the chemiosmosis. So the first requirement is a membrane. As I told you, the first requirement for chemiosmosis is a membrane, and second requirement is a proton gradient, as their water moved from a region of high concentration to a region of low concentration. Here also requires a proton gradient, and remember, proton gradient is H plus gradient between two regions. Then third requirement is ATPase. ATPase is an enzyme which comprises two parts. The two parts are F0 and F1. Now to describe F1, it is called the headpiece. It is globular, and also it is a peripheral membrane protein. Whereas F0, it is like a stalk or like a channel. It is an integral membrane protein. We know in the plasma membrane there are two types of protein, some peripheral and some integral. So this F0 is an integral protein and F1 is a peripheral protein. This F1 is also called head and F0 is also called stalk. Now the function of F1 particle is that there are the raw materials for ATP. ADP and P are present. So it's a site where ADP and P combines to form ATP. So the function of F1 particle is it is the site for ATP synthesis. Whereas the function of this F0 is it serves as a channel for passing of the protons. Okay, the protons passes. See, the first proton passed, second proton passed, third proton passed. So it's a channel through which the proton passes. And remember, when the proton passes, will this reaction as ADP gets coupled with P to form ATP. So it's very, very important that the proton pass. Only when the proton passes and reaches here, ADP couples with P to form ATP. So this is the structure of ATPase. So once again, ATPase comprises two parts, a globular head which is indicated in green and a stalk. The globular head is the F1 particle and the stalk is the F0 particle. Both are proteins. This is a peripheral protein. The F1 is a peripheral protein, whereas F0 is an integral protein. Now the F1, the function of F1 is it's a site for ATP synthesis. That is where there is the raw material ADP and P. Whereas this F0, the function, it is a channel through which the protons passes, and only when the protons passes, see if I indicate protons with yellow, only when protons passes will the ADP get coupled with P to form ATP. That's why it's very important. And the last requirement is ETC or the proton pump. ETC or the proton pump is a chain of electron carriers. These are indicating some electron carriers. Okay, let this be electron carrier 1, electron carrier 2, 3, 4, 5, 6, 7. So, in this chain there are electron carriers, and these electron carriers functions to pass the electrons. Electron just passes from one carrier to another carrier, and during this passing of the electrons are not used, they are just passed. But one thing happens, protons are removed from one region to another region. For example, if I put it like in a beaker, and we want, like the raisins, we want the protons or water to move from the beaker into the raisins, which is indicated in this oblong structure. Okay, what should happen? There should be high water concentration outside than inside. Inside also there will be water, but look at the number of water molecules which is indicated here. Here there are 4 water molecules outside, whereas inside there is 1 water molecule. So there is a gradient. So how was this gradient created? It is not with water, children, it is with hydrogen. So I am removing that oxygen, it is with respect to hydrogen. So proton gradient is created, that is when the electrons, I am reiterating, when the electrons are passed from one electron carrier to the other electron carrier, what happens is a proton is removed. Where? In this case, proton is removed here in this space. See here, protons are added here. How? From where it is removed? So it might be removed from inside. When we say removed, it means it is from inside. It is removed and it's going outside. So that a gradient is created. So we have more protons outside than inside. So the purpose of an electron transport chain is for passing of electrons, and when the electrons are passed, remember, protons from one region are removed and added to another region. So here we saw protons from inside is removed and added to the outside. So when this happened, what happened? A gradient is created. Therefore we can call this ETC is justified by calling it as a proton pump, because of ETC a gradient was created. It's not ready-made gradient as in the water and the raisins, where water it is outside high water concentration, and inside definitely inside the raisins it's no water, because it is already ready-made. But here it should create that is created by the ETC. How ETC operates? By removing protons from one region and adding the protons to another region, so that a gradient is created. It's not equal high and low. Therefore, this movement of protons from a region of high concentration to a region of low concentration. So these are the 4 requisites of chemiosmosis: a membrane, a proton gradient, and we know what is responsible for proton gradient, ETC, and lastly ATPase is also needed. ATPase is also called F1 particle. Now having this in mind, I am going to draw the electron transport chain operating in the mitochondria as well as in the chloroplast. First let me draw the electron transport chain operating in the mitochondria. So now this is depicting the outer mitochondrial membrane. Next I am going to draw the inner mitochondrial membrane. I purposely gave a thick nib to this, because it is in the inner mitochondrial membrane that our ETC is operating. So rather than zooming the inner mitochondrial membrane to show the electrons, I made the inner mitochondrial membrane thick to show the location of the ETC. So once again I am going to label this. This is the mitochondria. the outer mitochondrial membrane indicated by the thin oval, inner mitochondrial membrane, and remember why I made it thick? To incorporate our ETC. So, ETC, a system or chain of electron carriers. So, I am going to 1, 2, 3, 4, 5, 6, 7. Okay. So, this is our electron carriers, and at the end of our ETC I am drawing something. See what is this? Our ATPase, and look how is the ATPase. It is having the stalk and the F1 particle, which is a globular head. Okay, see the way I drew it. So, everything as per the requisites are drawn. ATPase, and this is our electron transport chain, ETC. And definitely the two regions where the proton gradient is going to be shown. Which are the two regions? The center is the matrix, and this space between the inner mitochondrial membrane and outer mitochondrial membrane, that space is called the intermembrane space. Now, I will give you a clue where should the protons be accumulated more. The clue is looking at the globular head. Look at the globular head. Okay, it is projecting into the matrix, which means that where it is projecting into, there the protons should be less. Suppose there are 4 protons and outside there are 2 protons. It's not right. Outside should be having more number of protons compared to the inside. Okay. Now, I told you the electron ETC is only operating like a pump to create the proton gradient. So, as the electrons, I am indicating the electrons in blue color, it passes from one electron carrier to another electron carrier. As it is passing, what is it doing is, it is removing the proton from one region and adding it to another region. So, it came here. Again, this proton from the matrix is removed and added here. Again, this proton from the matrix see, the proton is removed from this region and added here. So, this is brought about by the electron transport chain. How? When the electrons are passing, see the electron pass. When it passes from one electron carrier to another electron carrier, what happens? The protons from one region, here the region is the matrix. See the matrix. It moves, the proton moves from the matrix to which region? To the intermembrane space. So, like that way, a lot of protons are added to the intermembrane space, and you can see that the number of protons inside is less. So, here in this case, we have only 2 protons, and count the number of protons in the intermembrane space, more 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Okay, so we have 12 protons in the intermembrane space, which is more than 2 protons in the matrix. So, a gradient is created. Then this will help. What? In ATP synthesis, we have the raw materials ADP here and P, but they should get coupled. How do they get the energy, or how will it get coupled when the proton gets in? So, the proton and carry, as it moves through the channel, see that F0 to F1, what happens? The ADP and P binds, see, and bringing in protons. See, protons added. So, you can see protons move from a region of high concentration to a region of low concentration. See, high concentration is the intermembrane space. Intermembrane space is the region of high concentration to a region of low concentration. And then what happens? This ADP plus P combines to form ATP. See? Clear? The same setup I am going to show in chloroplast. Here we are seeing the inner compartment. Inside the inner compartment, which is called the stroma, we have the thylakoid. We know what are thylakoids. We see the thylakoid stacked to form the grana. I am just exaggerating only one thylakoid. So, do not misunderstand, only one thylakoid is elaborated. Okay, giving a totally different appearance. Actually, in the stroma, there are a number of thylakoids arranged to form a stack called granum. So, now concentrating only on the four requirements, I am labeling these structures. This region inside the thylakoid is called lumen, and outside the thylakoid is called stroma. So, the two regions we have identified. And what will the yellow be, which I deliberately made thick? The thylakoid membrane. So, from that you can understand I deliberately made it thick to accommodate our ETC. So, I am going to accommodate our ETC. Same color I am using, red. ETCs, one electron carrier, second electron, third, fourth, fifth, sixth, seventh. Okay, same number I am putting. Electron transport chain with 7 electron carriers, and it finally ends up with the ATPase. Look at the way I am drawing the ATPase here. See the opposite direction. Where it is the globular head projecting into, it is projecting into the stroma. So, from this where we can guess the gradient. Where should the proton gradient be high? It should be inside the lumen. It should be high inside the lumen compared to the stroma. So, again I am going to show the working of the electron transport chain. This is the electron. As it is moving from one carrier to another carrier, what is happening? The protons from the stroma is removed. See the reverse way, and gets accumulated in the lumen. So, I am adding to the lumen. See, it is added to the lumen. Added to the lumen. I am not going to remove all the protons. It is not going to be 0. There will be some protons out in the stroma, but you look at the number of protons in the lumen: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So, there are 12 protons inside the lumen, but look at outside: 1, 2, 3, 4. So, 4. So, we have more protons in the lumen, say 12 more than what is in the stroma. So, proton gradient is created. Yes. And now what happens is, here there are the raw materials A, D, P and P. They have to combine. How will they combine? Only when our protons get in. So, our protons will get in. They will move from the lumen into the stalk, and then it gets into the globular head. Again the protons get in. See, they are passing through the channel. Again the protons passes through the channel and get into the globular head. Again protons from the lumen of the thylakoid gets in through the channel called the F0 to the F1, and this results in the coupling of A, D, P and P to form ATP. Okay. So, I hope you have understood both the diagrams and the purpose of the electron transport chain. Now, with these aspects in mind, we are going to see the differences in both the mitochondria and the chloroplast. This is only an intro video. When we deal with the electron transport chain in detail, this will be more clear. So, the first parameter we are going to look into is the ETC operating in chapter of photosynthesis, which is indicating chapter 13 is photophosphorylation, and the ETC operating in chapter respiration, that is L14, it is oxidative phosphorylation. So, this side is applicable for chapter 13, that is photosynthesis in higher plants, and this side, which is oxidative phosphorylation, is applicable for respiration in plants. Okay. So, the first parameter, where is photorespiration occurring? When is photorespiration occurring? It is occurring during photosynthesis. At the same time, when is oxidative phosphorylation occurring? During respiration. Second, where does photorespiration occurs? We saw it occurs in the chloroplast, whereas oxidative phosphorylation is occurring in the mitochondria. Next, where is the ETC operating? We saw in the thylakoid membrane, that only was made thick. You will never forget, thick yellow. Whereas in the mitochondria, where is the ETC operating or located? In the inner mitochondrial membrane. Fourth, where is the ATP is located? You saw the color I use is red for the electron transport chain. I ended with drawing up the ATP is also in red. So, it is in the thylakoid membrane. Here it is the inner mitochondrial membrane. Then we know where is the gradient created. It is in the case of the chloroplast, the gradient is created between the lumen and the stroma. If I draw, this is the stroma and this is the lumen, we saw more of the protons in the lumen and less in the stroma. Okay, so if I give a proton gradient is created like this way, more in the lumen than in the stroma, whereas here it is the more in the intermembrane space than in the matrix. Okay, proton gradient is created. Next, 6, that's the very same point, proton accumulation. Here I told you where the proton accumulation is in the lumen. Here where the proton accumulation is in the intermembrane space. Next, 7th point, ATP is produced into, there you must draw our ATPs. In this diagram, how did I draw the ATPs projecting out into the stroma, whereas here I drew the diagram projecting into the matrix. So, the answer to that, ATP is produced where? Into the stroma, whereas here ATP is produced into the matrix. The globular head is facing the matrix. Where it is facing? Here it is facing into the stroma, so it is stroma. Here it is facing into the matrix, so it is matrix. Then 8th feature, energy is used from light. Yes, because it is during photosynthesis, whereas here the energy is utilized by the oxidation of those molecules. Energy molecule NADH we know, as well as FADH2. Okay, so energy is from light when it comes to photophosphorylation, but when it comes to oxidative phosphorylation, it is due to energy is from by the oxidation of these two energy molecules, NADH and FADH2. And lastly, the role of water. When I deal with this exclusively, you will understand in photophosphorylation, the splitting of water, opposite of splitting is formation, whereas in respiration we have formation of water. I hope you have understood the differences. And please have these diagrams to correlate with the differences. So, a quick recap. Look, where is the ETC located in? If this is representing the chloroplast and this is representing the mitochondria, a few points which I can elicit from the diagram. See, where is the ETC located here? It is located in the thylakoid membrane, whereas here it is located in the inner mitochondrial membrane. Second, look at the ATPase, where it is located again in the thylakoid membrane. Here ATPase is located in the inner mitochondrial membrane. Then next, where is the gradient, proton gradient more? Here it is in the lumen, whereas here the proton gradient is the intermembrane space. Fourth point, where is the ATP going to be synthesized? Look, where is the globular head facing? So, ATP is going to be synthesized in the stroma, whereas here the globular head is facing into the matrix. So, the ATP is going to be synthesized into the matrix. So, many points we can elicit from the diagram, and also you should correlate it with the diagram, the differences with respect to the diagram. I hope you have understood these differences. Thank you.